Hi, today we're going to talk about the role of nucleic acids in gene expression with protein synthesis as our main focus. And this is just going to be an introduction, so we're not going to get too deep into it, but we want to review some important vocabulary and really the process, the steps that go into it. So let's dive in. Let's talk about language before the process, because you need to know these words to really be able to interact with the concepts. So we have DNA. This is the genetic material that stores instructions for building proteins, and it's found in the nucleus. We have RNA. RNA is the nucleic acid that helps carry out DNA's instructions. We have messenger RNA, or mRNA, and that carries DNA's code from the nucleus to the ribosome. We have transfer RNA, tRNA, and that brings amino acids to the ribosome during translation. We have ribosome, which we've already mentioned, and we should be well aware because we've talked about this organelle many times before. But ribosomes are the cell structure or organelle where proteins are assembled. Codon. A codon is a set of 3 nucleotides, so it's a triplet on mRNA that codes for 1 amino acid. So every codon has 3 nucleotides and the code for 1 amino acid. A peptide bond. So this is the chemical bond that links your amino acids in as they form a protein. So it's the bond. Something that we've talked about other bonds with before is that hydrogen bond, that weak bond that combines our nitrogen bases within nucleotides that gives us that double helix form. So this is a different type of bond, but it's a chemical bond and it links amino acids into a protein. Polypeptide. So this is a chain of amino acids that folds into a functional protein. A start codon. This signals the ribosome to start translation. So I like to always think of start codons as the beginning of a sentence. In English, we capitalize the first letter of our sentence and it lets us know, oh, this is the new start. We have stop codons, and these signal the ribosome to stop translation. So again, to extend that analogy, I like to think of these like a grammatical mark, like a period or an exclamation where we're done with that sentence. So, protein synthesis. This is just how cells make proteins. And we need to understand DNA, RNA, and the ribosome to really interact with this, which is why we've previously talked about ribosomes and DNA and RNA. So we should have all of these building blocks where we're starting to pull information together. How does the information in DNA become a functional protein? Well, let's think back to what proteins do. Well, proteins can be structural, they can be signalers, they can be enzymes. And we talked about enzymes a lot previously in our biomolecules unit. And enzymes, we use that lock and key model to think about them because enzymes have a specific shape and they're paired with a substrate that only matches at the active site on the enzyme, and they have to be exact. It's an exact match, like a lock going into, like a key going into a lock. Well, enzymes are just a type of protein, so all proteins have specific shapes. And shape is going to help determine their function or what they can or cannot do. So, protein synthesis. What is it? Well, protein synthesis is just the process by which cells make proteins. Why is it important? Well, you should have a lot of ideas as to why it's important, but let's think about the state of Texas and why CCISD thinks protein synthesis is important for you guys to learn. Well, proteins are essential for structure, enzymes, hormones, and cell functions. So it's pretty gosh darn important. So we want to know about this. Well, we want to know where does it happen and what transcription and translation are. Well, protein synthesis has two steps, transcription and translation. Transcription happens in the nucleus, and translation happens in the ribosomes, or rather at the ribosomes, which are in the cytoplasm or the rough ER. So remember when we talked about our organelles, ribosomes can be free-floating in our cytoplasm, or they can sometimes be attached to our rough endoplasmic reticulum. Transcription, and I have that C, that's not just a typo. I like to remember the stages or how the order of this happens by thinking of the alphabet. Well, C comes before L in the alphabet, so transcription with a C happens before translation with an L. So transcription happens in the nucleus, and what happens? Well, transcription is when DNA is used as a template to make messenger RNA. And this happens so that DNA, that code, can leave the nucleus safely to instruct protein production. Remember, DNA is in every cell of our body in the nucleus, but that DNA likes to be very safe. It's not going to leave the nucleus. It's sort of like playing a tabletop game, and if you get Australia, you don't want to leave Australia because it's much easier to win the game. So your DNA likes to stay inside the nucleus. So translation, remember L comes after C, so translation happens after transcription. It happens at the ribosome in cytoplasm or on the rough endoplasmic reticulum. What happens during translation is the ribosome reads the messenger RNA codons, and remember codons are those triplet nucleotide bases, those nitrogen bases. And transfer RNA brings amino acids, and the peptide bonds link them into a polypeptide. So the ribosome reads the messenger RNA codons. Every codon then is coding for a specific amino acid, and transfer RNA brings that amino acid to the ribosome. And once you have your amino acids and they start getting stacked, they need to be bonded together, and they're bonded by peptide bonds. And when you have a whole series of them, you form a polypeptide. So why does this happen? Well, translation happens to build the protein according to the DNA's instructions. That's all. So, start and stop codons. Start codons, this is primarily going to be AUG, and it's signaling the beginning of translation. Stop codons, UAA, UAG, UGA. Remember, I've talked about how codons can sometimes seem a little repetitive. Well, that's because our amino acids are somewhat redundant. We have different ways of coding for amino acids, and so there's some redundancy built in. And when we talk more about mutations, we'll see how we can sometimes have a mutation and it doesn't cause something bad or negative to happen because it's a silent mutation. We don't really notice it because it happens to code for the same codon or amino acid. So our stop codons, they just signal the end of translation. Why do we have start and stop in protein synthesis? Well, when the ribosome reads that messenger RNA, it has to know when to start building the protein and when to stop. Remember, that messenger RNA is copying sections of your DNA, and your DNA has all of your information. Well, you don't always need to use all of your information. So we want to make the correct protein. We don't need to make all of your DNA. We don't need to make a copy of all of your DNA. We're building one specific thing. So we're making a small package, one protein, not your entire genetic code. So we want to have a start and a stop. So your start and stop codons they act like punctuation marks in a sentence. So a capital letter tells you, oh, the sentence has started, and a period or an exclamation, they're going to tell you that the sentence has ended. So without it, the ribosome, it's not thinking, it doesn't have a brain. Your ribosome wouldn't know where to begin or end its instructions. So how do we get from a polypeptide to a protein? Well, the polypeptide folds into the functional protein. And this is happening because protein shape determines function in the cell. Remember, we talked a lot about the shape of our enzymes, and enzymes are a type of protein. But this specific shape is something that we talk about a lot with proteins. They need their specific shape. And that's because protein shape matters. The shape of a protein decides what it can do. Each protein has a special shape that lets it fit or connect with other molecules in the cell, just like our lock and key model that we already talked about with enzymes. But it's true for other proteins, not just enzymes. If the shape changes, the protein might not work correctly anymore. And when we talk about the mutation sickle cell anemia, that is a mutation that changes the shape of the protein. The one codon, instead of coding for glutamic acid, it codes for valine, and that one change changes how the protein is formed. And because it's not formed the way it should be, your hemoglobin ends up not being formed correctly either. So you end up with rigid C or sickle cell shaped blood cells instead of rounded, flexible blood cells, all because one amino acid replaced another. So we'll talk more about that specific type of mutation later in the week. Remember, your genes tell your cells which protein. The proteins are going to determine the traits. So the wrong sequence will have the wrong folding, and the wrong shape equals sometimes loss of function. So if the protein isn't folded correctly, you're not going to have the correct shape, and because you don't have the correct shape, you're not going to have the same functionality. So let's review, because this was pretty quick, and it was a lot of review though, so we could move fast through this. But let's make sure we get some of these important key features. DNA stays in the nucleus. Messenger mRNA carries the code out of the nucleus. Transcription is making a copy of your DNA, turning your DNA into a copy that can leave the nucleus, and that copy that leaves the nucleus is your messenger RNA. Translation is your messenger mRNA being turned into a protein, a polypeptide. Proteins are made of amino acids linked by peptide bonds. And you have start and stop codons that ensure proteins are made correctly. So how could we diagram this? Well, here's how I might diagram it in my notes. If I'm looking at a cell, and I'm going to draw an animal cell because why not? We have our amorphous blob shape for our animal cell, our eukaryotic cell, and we have a nucleus. And I'm going to make some ribosome dots because we have lots of ribosomes in our cells. I'm going to maybe draw my Golgi apparatus or my endoplasmic reticulum, because if you see here, the Golgi body and the endoplasmic reticulum can look similar. But if you stud it with ribosomes, you go, oh, it has to be rough ER. Well, within my nucleus, I'm going to have my DNA, which I'm going to represent with a double helix form, and I'll even try and make some of my nitrogen bases on the nucleotides to, really demonstrate that. From our DNA, we're going to, and we always like to label our diagrams to the best of our ability. So we have DNA inside the nucleus. Our transcription will happen with this DNA in the nucleus. So we need to do what? Well, we need to make a copy of this DNA because the DNA can't leave the nucleus, so we need to make a copy, a functional copy that can leave, and that's going to be a single strand of mRNA, messenger RNA, and that's going to, remember, when we're making RNA, we end up replacing our thymine with uracil, but it's still pretty similar. We've done this coding before. So our messenger RNA can get out of the nucleus, and because it can get out of the nucleus, we're going to then have it go to a ribosome. So here you can see that there's more labeling because even though we make our diagrams, we want to make sure we're labeling them so that we don't confuse ourselves. So we have messenger RNA. It has now traveled outside the nucleus. It's in the cytoplasm. And it has to get to a ribosome, also within the cytoplasm. And at the ribosome, it can now start the process of translation. Translation happens at the ribosome. And translation is where your codons, your triplets are going to be read, and each triplet, each codon will have the code for one amino acid, and your amino acids are going to start forming a polypeptide, and they're going to be bonded with peptide bonds, and these bonds are going to form and fold into a functional protein at the end. So I would diagram this if for no other reason than to really walk yourself through the process and show that, oh, I can do this. All right, hopefully this all made sense, and if it didn't, ask questions, email me, ask me during class. I love talking about it, and I can't wait to talk more about codons when we read our codon charts next week. All right, have a great day.